So I'm very happy to get to interview John because I owe him a lot. I owe him like a second life in mycology because he let me go to his lab a few years ago. But anyway, I'd like to start out by asking you about your family. Did your family have an academic background or a mycological interest? Or?
Okay, so neither. My family, my parents and the parents of my wife, Delia, grew up on farms. Mm-hmm. So they're the generation that left the farm and came to the city. Neither of them, none of them had more than a high school education. Although they had good high school educations, probably equivalent to a community college today. Mm-hmm. And they, my mother grew up on a wheat farm in Nebraska. My wife's mother on a apple farm in New York. Her father was on a cotton farm in Louisiana, and my dad on a homestead in Idaho. And that his mother, my grandmother, traveled in a covered wagon from Texas to Idaho. So I have an interesting background. But I grew up in Los Angeles, in a highly urbanized area. Mm-hmm. And I was not drawn to fungi. Some mycologists start out with an interest in fungi very early on. I didn't become interested in fungi until I was a senior at UC Berkeley, and I did a senior project with a very charismatic mycologist, Ralph Emerson. And that got me interested. That made me think mycology was a possibility. I was more interested at that point in plants. Mm-hmm. But I had started university with an interest in chemistry from high school, and I didn't have a good biology course in high school. He was a retired military man, and it was not a very interesting biology course. But chemistry and physics were really interesting. I got to Berkeley, took chemistry, did well, but I didn't like it. So I looked for the major with the least chemistry. That was physics. So I did physics, but I wasn't very good at physics. But I worked for the Boy Scouts in the summer at a Boy Scout camp. And there was a need for somebody to teach natural history, nature merit badge. And so that got me to take a plant identification course at Berkeley to learn the plants. And that was so much fun compared to physics, mm-hmm. which I wasn't very good at, that I decided to become a biologist. That was actually intellectually very difficult, psychologically, to give up on physics, but I'm obviously very happy that I did that. Yeah.
Yeah, when you mentioned Boy Scouts, it reminds me of something. Dean Glavy once was going to do a Boy Scout merit badge in mycology. Do you think
that would be a brilliant idea?
Is there anyone in the audience that might like to do that? Because that would get young kids interested. We could do Girl Scouts too.
Well, you may not know that in the United States, there's now the Scouts.
I know.
What was the Boy Scouts now, except girls and boys?
But they're still Girl Scouts.
But they're still Girl Scouts, mm-hmm. and so both groups could have a merit badge of mycology.
Yeah.
It would be very good. Yeah.
So it'd be a great thing for someone to do who needs a project. Okay, so you mentioned you did a project with Ralph Emerson. How did you come to do that? Was it a general biology course or?
No, well, there was the opportunity to do a senior project, and so I read about that, and I thought I should mm-hmm. do this, because I was thinking at that point, what am I going to do after I graduate? And I didn't really have any skills as a biology major, and I went to the university during the Vietnam War. And so if you stayed at university, you didn't go to war, and if you failed, you went to war.
Yeah.
And then
that's when our grade inflation supposedly began.
And so then, as graduation approached, I thought, what should I do next? And I thought doing a senior project would tell me, am I interested in this at all? And having worked in the Boy Scouts in the Sierra Nevada, there's a very interesting plant that's bright red, has no chlorophyll, and behaves like a fungus. It makes tiny little spores. So I went to the dean and told him that I wanted to do a senior project and sort of what it was about, and he suggested Emerson. So I went to visit Emerson, and I opened the door, and I talked to him, and he goes, I'm too busy, can't do it. And then he kind of goes, and he goes, come back in a week. So I came back in a week, and he said he would do it, and he had a postdoc, Carolyn Tarosian, who taught me how to do microtomy, how to make wax sections of this plant and look at the roots of the plant, which had a mycorrhizae. And that got me interested in fungi and microscopy. So when I went to graduate school at UC Davis, and there you got to rotate. Well, you earned a master's first, so you got to look around the department and see who to work with. And I was interested in plant taxonomy and also mycology, but Ken Wells, who was the mycologist, he was not a super charismatic person, but he was a really good person. And he had one student, and the plant taxonomist had dozens of students. So I thought, well, I'll be a mycologist because I really got along with Wells, and I'll get more attention than the plant taxonomist. But I did microscopy. Yeah.
Okay, but how did you happen to go to Davis when you were at Berkeley?
So I applied up and down the West Coast, and I got into every place I applied to, probably because I had done a senior project, and had a letter from Emerson. And the last university to accept me was Davis, which at that time had a very highly ranked botany department. It had a botany department. Yeah. And they offered me the most money. So I went to Davis.
He was bought. He was bought.
and they offered a fellowship where for 2 years that you had a fellowship, and then another 2 years you taught. And also I discovered, well, I knew from having been a Boy Scout that I like to teach, and that anybody who's starting out, any training you can get talking before groups of people, if you want to be an academic, that will tell you if you want to be an academic, and I liked it, and that training was really useful when I went to Davis and was a teaching assistant.
Did you meet Delia there?
I met Delia at Davis. Yeah, I saw her several years before we actually met. And it took a while for us to get together, but eventually, yeah. Okay,
so then we jumped to Georgia?
Yeah, well then mycological congresses. I went to my first mycological congress was in Tampa, Florida. And I went there, and I was an electron microscopist. Ken Wells did electron microscopy. And he let me flounder with a snow plant project for a year before saying, this isn't working. And he had grant money to look at nuclear division in Basidiomycete yeasts, which I did for my thesis. And I went to Tampa, and the great hero of mycology, Manfred Gerbardt, who was an East German electron microscopist and light microscopist, who was fantastic, was there. I got to meet him. And I met Mel Fuller, and he looked at my poster and offered me a postdoc. And this will sound crazy, but this is 1977, is that right? I think. And there were 3 opportunities for postdocs in the United States in mycology. There was Bartnicki Garcia at Riverside, who had an electron microscopist. There was George Kobayashi at Washington University in St. Louis, a medical mycologist. He wasn't interested in me. And Mel Fuller at Georgia. And so Fuller said yes, and so then then Delia said, I'm not going to Georgia as your girlfriend, so we got married. And then we went to Athens, and I worked with Mel and did electron microscopy of chytrids. And but okay, this is very interesting. While there, Delia worked in a DNA lab. One of the first people to look at plant molecular development, molecular biology, Richard Meager. So I'd go there for lunch and talk to people about working with DNA. And there was another person in the genetics department, Robert Landsman, who had convinced John Avis to look at mitochondrial DNA in animals. And so I listened to him and thought,
I didn't realize Avis had started that early.
Yeah, that's what I thought that's the future, because I was interested in evolution. It took a year to do the electron microscopy of one fungus. And then when you were through, you compared that information to work that other people had done, and they never did it the way you did it. So it was hard to compare, I mean, people weren't doing that. And I looked at the DNA and thought, this is going to be the way. So when I got to Berkeley, it took a while, but I switched.
I remember seeing the job ad, and we all wondered who in the world will be able to fill that position, because they demanded so much in the job ad.
Job ads always demand.
They wanted, I think you even were supposed to do something at the botanical garden and keep the collection going, the fungal culture collection and the herbarium.
Yeah, still is a fungal collection called the Micro Garden, and Emerson started that, and it's a teaching collection. And he argued with the administration that look at the money you put into the botanical garden, you have to put some money into the microbes. And it's been hard to keep that money. We still have a technician part-time, but it's not as good as it was when I got there. It has had some tough times.
So you got there and you didn't do any microscopy?
No, that's not true. So Mel Fuller was a great postdoc advisor.
Oh, I have to, Mel Fuller once gave me advice when I went to meetings. I was told not to wear blue jeans. Mel Fuller told me that.
Yeah.
So that was a different kind of lab you were in.
Mel Fuller gave you advice about life. He did. He was really good. He also would, tell you when he thought you were, doing something wrong. So in fact, he told me I had to do electron microscopy his way. And I just, I thought, well, I have a good way to do it. So I did it both his way and my way, and developed the photographs, put them on a big piece of cardboard that had been the box for a refrigerator, and asked him which photos did he like, and he picked mine. I went, those are mine, and he went, okay, and then he didn't give me.
And I want to ask one question about Mel, and then I'll quit and let you go on. But did he have his loom in the lab at the time?
Okay, so Mel is an entrepreneur. I don't know if you know this. He had been a professor at Berkeley. He was one of Emerson's students, and will Mel see this video?
No,
oh,
no,
okay.
no. Mel's quit mycology.
So Mel, being a Emerson student, although he was a professor, he was still Emerson's student. That's the way he felt. Yeah. So he got out of there, and he was offered a very nice job in Georgia. And he had a house on the hill in Berkeley, which he sold to an incoming faculty member.
Russell.
And bought a house, Russell Jones, and bought a house in Athens, Georgia, and a shop for weaving. And so he had first he bought a nursery. He had a plant nursery, and that did well, and then he sold that and bought a weaving shop in Athens. So Mel
was,
yeah.
Anyway.
So when I got to Berkeley, I knew I wanted to do work on DNA, but I didn't know how. And Ralph Emerson, very sadly, had died of lung cancer. I think maybe just as he retired, or maybe he hadn't even retired.
I remember at MSA meeting, there was a big workshop in his honor, and he missed it.
That was in Athens, in '78.
Died right after.
And so, but his last student was Don Natvik, and Don was an expert in fungal physiology for his PhD. So Don and I started a talk, because Don didn't have a postdoc, and I hired him at slave wages. I had a grant, an electron microscopic grant, that Fuller had let me write at Georgia and let me take with me to Berkeley. And so I had some money, and we started to work on DNA, and we worked on mitochondrial DNA because that was a small molecule, and the animal people worked on it because it was haploid. They didn't have to deal with diploids. And so we got a zoologist at Berkeley, he and his postdoc, Olin Anderson, taught Don and me how to clone DNA, and we started looking at mitochondrial DNA and using it to try to understand fungal evolution. And we did that, and I remember going to the MSA meeting in 1984 in Fort Collins with a mitochondrial DNA map of Neurospora and some very simple studies, and thinking, this is gonna, nobody's gonna have anything like this. And Tom Bruns was coming there from Michigan with this exact same study with Swillis, thinking nobody's gonna have anything like this. And we got there and met each other and plotted for his next step, would he come to Berkeley as a postdoc, which he came would be 88, I think. It took a couple years for him to finish it. And he wrote a successful NSF grant that funded his postdoc visit to Berkeley. So Don Natvig and Beverly Smolich was a technician, and those very early days. And Georgiana May was my first student, and she had been a master's student at Georgia. And she then, when I came to Berkeley, she applied to come to Berkeley, and the department admitted her as a student. So that Don and Georgiana and Bev Smolich and I, that was the group that started.
Yeah. And then what happens next? When does PCR come in?
Okay, so that was, I got there in 80. In 82, well, when I got there, Ray Collins was the chairman. I don't know how many people remember Ray. He studied myxomycete genetics. And he also died young. So the history of mycologists at Berkeley, their longevity is not a good one. And that's part of the reason I retired.
I was going to
For the first few years there, I lived in a building that the air handling system was not in good shape, and we always wonder. Anyway, so Ray, when I got there like 3 weeks before the start of the semester, or a quarter then, and I was teaching with Ray, and then he said, by the way, you're in charge of the colloquium. And I said, well, oh, that's nice, what's that? Oh, that's the seminar series. Oh, and when does that start? In 3 weeks. And they had invited no one.
No
you know? And so I said, okay, well, what's the budget? There is no budget. So I looked at everybody who studied fungi or botany within about 50 miles of Berkeley to invite them to come and give talks. So UC Davis, I knew people there. I didn't, and San Francisco State, Harry Tiers. And I didn't know people at Stanford, but there was a mycologist there, David Perkins. And so I remember Perkins came, and Natvig and I, Don Natvig, were talking about, yeah, okay, we want to study molecular evolution, but what fungus? And Perkins talked about Neurospora. And we just looked at each other and went, oh my God, this is the thing to look at. So that's how we started doing that. Then there was one of the first biological startups, period, I think, was CETUS. And it started in Berkeley, and then it moved to a city next to Berkeley, Emeryville. And somehow I heard that there was a person there, a project there to turn cellulose into sugar. Sounds like biofuels, but this was actually sugar because the price of sugar had gone way up. Standard Oil had given them money. And there was a guy there, Tom White, who was in charge of that project. And he had cloned a fungal ribosomal gene. He was an Alan Wilson student. So he was very interested in evolution, and he had a technician, so he had her do that along with the genes that coded for enzymes that broke down cellulose. So I heard about him and invited him to come and give a talk just to the mycologists at Berkeley. So he came in 82 and talked about this, what they were doing in this gene, and we said, okay, we should do something with that. So we used it as a hybridization probe against green algae and red algae to see if red algae were closer to fungi than green algae, because you may remember there was a hypothesis that red algae were actually fungi.
The Floridian hypothesis.
The Floridian hypothesis. And this showed it disproved that hypothesis. And so then it took us several years to write that up, but we actually published that in '86. And so then, meanwhile, Tom and CETUS were developing PCR. Kerry Mullis was on the staff there, and Tom White was his boss. But we didn't know anything about that. And so I went on sabbatical in '88 to Sydney, Australia, to work with John Pitt. And this is also, this comes into another theme, but that we may get to, luck. Okay, yes. But Tom Bruns had come as a postdoc a month before I went to Australia. This is a good strategy. You hire, and I did it with Dave Geiser too. You hire a really good postdoc, and then you go on sabbatical. So you just give him the keys and go take care of things. So I'm in Sydney, and I get a fax from Tom White. And he says, CETUS has given me a sabbatical. I'd like to do it in your lab. I've got something I think you're going to like. Okay? And so in Australia at that time, every person had a fax machine. Okay? in Berkeley, the entire University of California, Berkeley had one fax machine 3 blocks from campus down Telegraph Avenue. And so I sent a fax back to the department chairman, Russell Jones, and said, do whatever this guy wants. And then I sent one to Tom, do whatever he wants. And Tom Brent sent one back, I already have. And so then Tom White came, and this was very early days of PCR. And our machine was Steve Lee, who's a graduate student. Three heating blocks, and Steve would move the tubes between the heating blocks.
I have a photograph of one of your early PCR machines. I'm not sure it ever worked, but I think you had a washing machine.
There was a washing machine too, yeah. So they had washing machine valves. It was the, Higuchi Hyundai or something. And it didn't work. We got it from CS because it didn't work, right? But then shortly thereafter, we actually got machines. And the early machines weren't very good. You had to use a thermocouple to figure out which little spots were actually good in the machine. But then, so then we started that. And all of the work that I did from, say, getting to Berkeley in 1980 or so up to 88 or 89 when I got back from Australia, none of that has been cited much. Okay, my thesis, not cited. Postdoc, not cited. In fact, you could put all those papers together, and it's a little bit of a stretch, but you could probably put all the citations of all those papers together, and they wouldn't count for my H-index, together. But they prepared us for PCR because we knew what to do with it. Yeah. And as Pester says, chance favors the prepared mind. He really said chance favors only the prepared mind. So I think that wasn't, it was fun doing all that work. It didn't have much impact, but PCR definitely had an impact.
As an aside, I lived 3 blocks from the high school where Carrie Mullis went to school. And I think in his Nobel address he said that his chemistry teacher gave them unfettered use of the chemistry lab.
Yeah.
Anyway.
Yeah. So luck, luck plays a huge role in life in general, and certainly in looking back at my career, the luck of getting to be a Boy Scout camp counselor. The leader of that group reached out to me, and I couldn't go one year, and he pestered me to go the next year. Or Ken Wells, or Ralph Emerson. And then, the luck of my wife working in a DNA lab, and then, meeting Tom White, and him having a fight with Cetus. Cetus didn't think PCR was worth anything. And he said, no, this really is worth something. And after the sabbatical, Cetus sold the rights for PCR to Roche for $300 million, and Roche hired Tom White to run their West Coast operation. So it's a chance is a big part, and no matter what how big your ego is, you have to remember you're lucky. Chance is lucky. Mm-hmm.
Well, I remember after you got all the primers done and everything and knew what you were doing, you let me go to your lab. And I was there for 6 weeks, I think. I lived in the Durant Hotel, which has now become Graduate Hotel.
I think it's, no, I think it's,
I keep getting these emails from
them. Okay.
But anyway, the thing about it was that you had the primers, and they weren't even published yet. So tell me about that paper.
Okay, so we, you have a choice when you have something new. Hold on to it and do as much as you can, or get them out. And we voted to get them out. And that was actually Tom White. That was his mantra too. And that proved to be the right thing to do because, science is a movement, and as it's creeping forward, things hold it back. So it gets held by something. And that was something that was holding things back. And so by putting those out as fast as possible, I think mycology really made use of molecular evolution more than many other groups, mainly because our things are so small, they didn't have many characters, and so we needed characters. But putting those out, those primers out fast was a good thing. And we let anybody who wanted to come, we would show them how to do it. And that proved to be a good strategy, I think, for the field, but also, this paper was mentioned yesterday, that it's now at almost 25,000 citations. And if we'd waited, other people would have developed primers, and there would have been competing papers to cite, and it wouldn't have been cited as much. So I think it was absolutely the correct thing to do.
Yeah.
And also, the primers were made well. And so Tom White did the 18S primers. Tom Bruns did the ITS primers. So.
And Tom at one time said, it's fast, and he wanted to call this business of collecting the ITS. He very, very early decided that would be a good molecule.
Yeah. Well, we wrote a annual reviews, and if you look back at that annual review article, it was predicted pretty well how things were going to go.
Yeah.
So this is another thing to think about. Okay, so science is a movement. You make a big contribution, so that was a big contribution. The bigger the contribution, I think, the shorter period between your making it and somebody else would have made it if you weren't there. So, Darwin and Wallace, or, Einstein, and he was collaborating with other people that had the same ideas. So that's kind of a sobering thought. If you didn't exist, the most important things that you've done as a scientist have been done by somebody else shortly thereafter. If you want to have something that lasts forever, do something really obscure that nobody cares
we
about.
talk about the time being right, but there are people, I think, who have failed, really. Not that they failed, but their contributions weren't recognized because no one came along right away.
Yeah, there are some. I think that's true. But I think it's, that's another thing to remember, to keep your ego in check, is that it's a movement. Science is a movement. It's not individual people, really. I mean, what's your contribution then? It's training people, I think. Because the more people you train, the greater chance one of them is going to make a contribution. And that also, another real piece of luck is getting a job at Berkeley. So when you get a job, you have to look good the moment that job opens. There's a tremendous amount of luck involved there. And you get a job at a place like Berkeley, then postdocs, walk in off the street. They come to you sometimes with their own money, or they're willing to come from another country. People like, Matt Fisher, D. Carter, people that left their home to come there.
Do you have a number in mind of how many people, do you know how many people have been trained as postdocs?
Oh, but postdoc and students, about 40.
40.
Yeah. And many of them have got jobs. Well, they've all got jobs, but many of them in academia. And some, somebody asked about regrets for a previous interview. And the regret is the really good students who then had some hard life situation and weren't able to take advantage of their talents.
Yeah.
I think that would be my regret. So
you had this wonderful job. You taught all kinds of people the methods and how to think, maybe. Why are you leaving it?
Okay. So I'm 68 years old. I'm not as good as I was when I was 30. And you know that. You're just not as quick, and also you can get tired more easily. The reason I retired at this point was that if I kept teaching, the university would not hire another mycologist because they don't need to. And so by retiring, there was the possibility that they would. And I knew that the department was putting up a request for a mycological position as their #1 request. So that was the moment to do it. And it might not have worked, but it did. So Berkeley, the administration has said that that the department will get a position for a mycologist. Now we don't know what kind of mycologist, but that's their problem. The old person shouldn't make those decisions. You want the young group to do that.
Yeah, I think we all noticed that there is a mycology job at Berkeley in Tom's talk yesterday. So anyway, so what will you do? Do you have hobbies?
Well, okay, so my retirement is a very soft one. So Berkeley has a avenue called a professor of the graduate school. It used to be a professor in the graduate school, but that acronym was unfortunate. And so they changed it from PIG to POG. And so what it means is I get my office and enough lab space for my people until I don't need it. And so I have a grant from the Department of Energy looking at the ecology of sorghum plants as they go through experience drought. And I have a fantastic postdoc, Cheng Gao, speaking of people who are willing to leave their country and actually an assistant professor job and come to Berkeley with his wife, and he's a tremendous, tremendously good at statistics and computational biology, and it's a big project. And so it's fascinating. The people are looking at the plants, they're looking, and we look at the fungi in rhizosphere, roots, soil, and leaves. And because we have so many data, we have weekly samples over the whole growing season with replication, 6 replicates. So we're really going to have some really interesting ecological output from it. So that's one project. And then I have also some money from the state of California to look at a fungal disease, Coccidiotomycosis. And this is another thing to think about. I've worked on Neurospora, which is, well, David Ketcheside is here, which is an absolute model organism. But I've also always worked on something practical. And Coccidioides, the human pathogen, is a practical thing. And you would think that you would do the first work in Neurospora and then apply it to Cocci. But often it's been the other way around. That the pioneering work on what's a species or what's the reproductive mode of fungi that you can't get to mate was done with Cocci, because 2 reasons. You can get the money to work on a pathogen more than a model often. And secondly, you can get the cultures, because physicians culture the fungus from their patients. No cultures, can't do population genetics, population biology. So I've got some money with Cocci, and what I'm interested in there is sampling in the environment to find out where Cocci is. So for this sorghum project, I go down south of Fresno, about a 3-hour drive from Berkeley, to sample the plants. And we don't do it every week, but we go down during the summer. Cocci, I've been going down every month for a year, sampling soil, and I have air samplers out, passive air samplers. And those samples are just now going to be looked at to see about the distribution of Cocci associated with small mammals. I think it's really a small mammal organism, both in the soil and the air over here. So that I'm very interested in. And I'm very interested in indoor air and asthma. And I haven't figured out how to get the money, but I have a good way to sample it in people's homes. And I would love to do that, because the asthma studies that are done with fungi now, all of the studies, this not just a few, have the sampling is 1 minute of vacuuming dust in the person's house once in their life, and they try to make a correlation between that and asthma. So we can do much better with that.
Yeah.
So I'm become an ecologist. Brens gives me a lot of trouble about that. But if you look at this meeting, everybody's become an ecologist because you can now sample the environment. And so it's the new microscope.
Yep.
You can look at fungi that we could not look at before.
Thank you. Does anyone have questions for John? Oh, question.
Dr. Taylor, you mentioned you collect dust samples. Traditional, you collect 1 minute. For you, sampling method, you collect for a month, use the petri dish or some kind of a plate?
That's a good question. So what we do, this was Tom Bruns's mantra, okay, that you have to have lots of samples. So it has to be inexpensive. And so his first vision was a pizza box. You'd open the pizza box. So that was a little unwieldy. So we started with round petri dish bottoms with nothing in them, and just set them out or hung them. And those took a month, okay? So the big advance was to use square petri dishes and hinge them so you could open them. Now you have 3 times the surface area. You could leave them out for a week. And a week is the minimum, I think, because it has to go 24 hours, because as we know, the spores in the air change throughout the day. And in the built environment, it has to go through a week, because people use the indoor environment differently during the week and during the weekend. And so if you can do at least a week sampling, then you get an idea of the fungi that are really there. Then you just swab it out and extract the DNA and sequence it and see what you have. And there's a talk today, there's a session today on indoor air that Joan Bennett organized, and one of my graduate students, Iman Sylvain, will talk about this in a housing project that's suffered water damage. So most of the studies are done in well-maintained buildings, and it's hard to get into a poorly maintained building because the landlord doesn't want to know. So you have to work with the occupants.
One quick question.
I hope it's quick, John. You played a pivotal role in the whole process towards one name for one fungus, one name. And now there's a very active, ongoing debate as to whether we should put names on fungi based only on DNA sequence. In a very short sentence, what do you think?
It could be one word.
So I'm going to give a talk on that on Saturday in a session on what's a species. And so that will be the long one. But the short one is, we have to do something about that, I think, now. And looking at the history of changes in taxonomy and nomenclature, we just have to do it. And there'll be a period to figure out how to do it right, and then the code will catch up.
Okay, well, thank you, John.
Thank you.